Okay, so we're now on chapter 20. And chapter 20 is all about sales. You should hopefully remember this from 7th grade. DNA was introduced to you in 7th grade too, but most of the time people don't remember 7th grade. So, hey, you may not. But first thing we want to talk about is characteristics of life. What makes something living? And so there are 5 characteristics that actually let you know that you are alive. One is that you have some type of metabolic process that takes up nutrition, processes, and eliminates waste. If you're alive, there is some type of growth process or regenerative process. You grow and you reproduce. If you are alive, you have a responsive process. You have some stimuli that you will respond to. You can adapt. Population will adapt or evolve. And then you have some type of control process, coordination and regulation of metabolic activities. And lastly, you have structural similarities such as cells. And so I always say a rock can't do any of these things, and that's how we know that a rock is not alive. You will not see a rock taking up nutrients and eliminate them. You won't see a rock growing and reproducing. You won't see a rock shiver because it's cold, meaning responding to a stimuli. You won't see it have any metabolic processes, and then it does not have cells. And so this comes in the form of a question. All these are characteristics of life except. So if you see that, pull this up and say, okay, they're on the 5 characteristics, which one is not present. All right, so the cell theory came about by Antoine van Leeuwenhoek. So he saw them and he called them animalcules. And then Robert Hooke came along and looked at them and gave them the term cells. And so those are 2 important names when we talk about cellular structure. There are 2 types of protoplasm that were later identified, or things inside the cell. One was the cell nucleus, which is the central body. It has fluid in it. And then you have cytoplasm, which is also known as protoplasm, and it's the fluid within the cell. So the cell has fluid that everything floats in, and then the nucleus has a fluid as well. Anything that we see inside of the cell are known as organelles. And so that term came about once we got better microscopes and we began to see that the cell just didn't have fluid known as cytoplasm or a nucleus, but it had other things. So you should know there are 2 major types of cells. You have prokaryotic and eukaryotic. Prokaryotes are usually your bacteria. Eukaryotes, there are 5 different categories: algae, protozoa, fungi, plants, and animals. So they both have common traits: cell membrane, cytoplasm, genetic material, meaning DNA or RNA, energy transfer molecules, which you'll find out are mitochondria or chloroplast, and then enzymes and coenzymes. So the thing here to remember is that you have 2 major types of cell: prokaryote and a eukaryote. The difference between the 2 is eukaryote is known as true nucleus. It has a nucleus. Prokaryotes do not have a nucleus. So somewhere right now, write down prokaryotes, no nucleus. Eukaryotes have a nucleus. So one of the things they have in common is the cell membrane. And the cell membrane is going to be composed of phospholipids and proteins. It is called a phospholipid bilayer because, as you see here in the figure, you have these heads, and then inside are tails, and then on the bottom you have a head, and inside are tails. You can look at the figure hopefully more closely on your computer, but just to show that you have these lipid molecules with heads and tails, and they come together to make this lipid bilayer. So as it says, you have a hydrophilic or water, philic loving head, which are on the outside. This ball here and this ball is here. These are the heads. And then you have fatty acid ends or hydrophobic tails. Hydrophobic, hydro, again, water, phobic or phobia, fear. So water-fearing tails are in between. So that means that the tails do not want to touch water and the heads like to touch water. So located on either side of the cell membrane are proteins, cholesterol, and carbohydrates, as you see here in the figure. And this is what gives the cell membrane a mosaic look. You know how you do mosaic tiles? So someone looked at the microscope, under the microscope once before, and saw it and called it a fluid mosaic model. So sometimes you'll hear that terminology used when we're talking about cell membrane. And I share that with you because every professor calls it what he likes to call it. Some will say cell membrane, plasma membrane, or fluid mosaic. Either way, we're all talking about the same thing. So when you have a cell, you have to have diffusion, meaning you have to move things back and forth across the cell. And so the net overall movement of a molecule from a place of high concentration to a place of low concentration. Diffusion is movement of a molecule from a place of high to a place of low. It's a passive process, must be no barrier for the free motion. Rate of diffusion is related to the kinetic energy, which you all remember, the movement, how much kinetic energy it has, the size, the concentration. The way I like to describe diffusion is when you move from a very packed room because it's hot to a room where there are fewer people because it's cooler. So that's moving from an area of high concentration of people to an area of low concentration. And the molecules keep doing that, moving from high to low until they reach what is called dynamic equilibrium. And dynamic equilibrium means that we're just equal on both sides. That means now that you've moved from a room where it's 20 people to a room where there was only 5, and the people keep moving from the room of high concentration, meaning a lot of people, until there are 10 or 12 people in each room. I should have said 24, so you have equal. But you're looking for dynamic equilibrium so that you have equal amounts on both sides. So osmosis is a process of diffusion, is a type of diffusion, and it is the movement of water molecules. All right, so let's make sure we know what we said. Diffusion was the movement of molecules from area of high to low. Osmosis is the movement of water from an area of high to low. And with this, you have to use a selectively permeable membrane. So when we say something is selectively permeable, it just means that it doesn't let any and everything in. So it's like that special club where you have to have a certain card to get into it. Like Sam's Club, right? And so without the Sam's Club card, you can't get in, you can't cross the barrier, or you can't cross the selectively permeable membrane. So when a cell is selectively permeable, it lets water in or out. When this happens, a cell can have 3 different things happen to it. It can either take up water, it can either expel water, or it can be in equilibrium with the water that it's in. So if you have an isotonic solution, meaning you have the same amount of water on the outside as inside of the cell has equal surroundings, so it's called isotonic. All right. If you have something called a hypertonic solution, meaning you have less water on the outside, then what will happen is that the cell will shrink. Why? Because the cell wants to lose water to put more water on the outside so that it can create equilibrium. Remember, we're always trying to create equilibrium. So in a hypertonic solution, the cell itself will shrink. In a hypotonic solution, where you have more water on the outside, less water on the inside of the cell, water will move in because it's going from area high to low, and the cell will begin to swell or burst. In science, we call bursting lysing or lysis, Lysis. If we were in class, I can write it for you. So it's called lysis or beginning to lyse. And because I am who I am, maybe you can see that. Lysis. All right. So when you're thinking about a cell, say for instance, someone who is diabetic and they have, as my grandma would say, sugar in the blood. Well, if you have a lot of sugar or glycosylation on your red blood cells is really what it is that then that creates sludge. So you have less water. And so this is why a diabetic is always thirsty, is because their cells are constantly giving off the water that it has to try and thin out the sugar in the blood. And so that means the cells are constantly shrinking because they're always giving out blood. And so a person who's diabetic always wants to take in water to try and create a balance so that the cell can have water and then there'll be more water within the blood. And so that's just an example of how this particular isotonic, hypotonic solution actually makes sense in real world scenarios. All right, so we talked about diffusion, osmosis, both are passive, and we want to talk about facilitated diffusion. And it's the same thing, moving from an area of high to low. It's a passive transport. The only thing that's different here is that you have to have a specific carrier protein, which is this blue membrane protein that you see here. And so that just means that you have help carrying your molecules from one side, the area of high concentration, to the other side, the area of low concentration. So facilitated diffusion only means that I have a carrier protein with me. Active transport is different. Instead of moving from high to low, you're going from low to high, meaning you're pushing against. Therefore, you have to expend energy. And so in active transport, energy is required. Whereas in the others, you only had no energy was required because it was passive. So in active transport, you also end up needing a carrier protein to move things from an area of low concentration to an area of high concentration. This is like you want to get into a crowded room and you have to push your way through, and the energy you have to expend to get into the room. It's the same thing that happens with active transport. A lot of energy is expended to get to the other side. Phagocytosis is another process of the membrane. It means that the cell membrane will actually wrap itself around a particle and engulf it. And so this just shows you the process of how that happens. So just know that phagocytosis is also a function of the membrane, which allows it to engulf or take in material on the outside of the cell. And so the membrane structures that we have in the cell are our cell membrane, which we just got through talking about. Nuclear membrane, it surrounds the nucleus. Endoplasmic reticulum, known as the ER, it can be smooth or rough. These are two things you should know. Smooth ER has no ribosomes, and the rough ER has ribosomes. And I believe that might be a question. So if you want to rewind this and hear it again. But smooth ER has no ribosomes, and the rough ER does. Then you have the Golgi apparatus, which I call the UPS of the cells. It is the flattened smooth membrane sacs that actually package and release certain molecules. So I like to think of it as the Golgi sees molecules, package them up, and put the address on it and says this goes to the heart, this goes to the liver, this goes to the spleen. All of that happens in the Golgi. So hint is it's the UPS of the cell. And then lastly, the membrane you have is a lysosome. And a lysosome I call the garbage man of the cell, because in the lysosome you'll see that it has these hydrolytic enzymes that actually break down debris back into the macromolecules that we can use. So lysosome, garbage man of the cell, he breaks down everything back into something usable. Golgi apparatus, the UPS of the cell. And I say that because I have to give you the hints that I usually use on the exam. And then inside the cell, this is just showing you. One thing I wanted to point out here is just penocytic vessel, which is a vesicle used for carrying water. So cells can eat, which we call phagocytosis. And then they can also drink, known as penocytosis. All right. So I'll end this right here.